Cool. Hey, Tariq. Thanks for having me.
Thanks for being here. I'm super happy to have you. It feels like our old lab meetings.
Yeah. I think the last time I was was it 2018, I guess? Was it COVID or before COVID? Maybe after
No,
COVID.
it was
During
during
COVID.
it. I think
You were in
yes.
Montreal, right? Yeah. I see.
I was in many places. I was in Montreal and I was also in Boston.
Like Boston as well with Chloe, if I'm not
Yeah.
mistaken.
Yes, exactly. I was in Boston and then I moved to Montreal, but still working with Chloe, then moved back to Boston.
I see.
Yeah, I mean, it was an interesting time. For everybody. Yeah.
All right.
Nice. Could you like to introduce yourself?
Yes. So my name is Mo Lotfollahi, currently a faculty here in UK in Wellcome Sanger Institute and also University of Cambridge Stem Cell Institute and then Cambridge Center for AI and Medicine here in University of Cambridge.
Hey everyone, this is not an ad. Go to blueword.com/in, that's BLUWR.com/in, like inside, and write about something that makes you think.
And I started my lab a year and a half ago, a year and a half ago. And we've been lucky and we've grown 15 to 18 people right now. Before I started this lab, I was doing my PhD in computational biology and machine learning in Munich with Fabian Theis. And before that, I was my background. I was trained as a computer scientist, and then like my master was in machine learning and AI, and I had no clue about biology. And I was actually I was just kind of like randomly taken toward the bioinformatics and computational biology because like back then I wanted to just like hardcore ML. And then I had this course in bioinformatics when I was doing my master's back in Iran. And I got into, I just like got exposed to bioinformatics. It was like old school bioinformatics, like I don't know, sequence alignment, like very algorithmic, I would say, rather than ML. And but I guess like just knowing how people analyze genomics data, and then it was this quite fascinating, and it's just like that kind of like drove me to further pursue this. Then I started my PhD in 2017 in Germany, 2017, 2018. And then during my PhD, most of my work was about the question was whether we can actually predict cellular responses to unseen drugs or unseen genetic perturbations. And so in late term, it's basically this technology, single cell genomics, allows us to extract molecular data. So basically information inside every single cell. And back then was quite expensive to do, but now it has been commercialized, but yet it is still expensive for most labs if you want to do it at scale. When I say at scale, if you want to, for example, if you have a disease sample and if you want to test thousands of drugs, it's kind of not possible for many places oral. And because of this, we need AI models, machine learning models, generative models, in general to potentially learn and understand this data, but most importantly, predict unseen experiments. Right? When I say unseen experiments, let's say you have a disease sample, and then you don't know how to reverse the disease toward the healthy because for example, you don't know anything about the mechanism of the disease, or you don't have a drug target for that, and that's quite closely aligned with a way of drug discovery called phenotypic drug discovery. which is you only have seen the phenotype, but you don't know what caused that or how it was caused, and you just want to reverse it. And that's why people do these high throughput screens. But usually when people do high throughput screens, what they measure is let's say a single gene, or they measure if cells die or they grow after a certain treatment. And then going beyond that, people do bulk RNA-seq, which is kind of an average version of single-cell RNA-seq. So you don't get the single-cell resolution, or you don't get the individual resolution of the molecular data for every cell. You just get an average information. So then, let's say if you have cells from your brain, and you have neurons, astrocyte, glia, and others, they kind of all collapse as an average effect. So you don't know whether the drug affected this cell type or that cell type. And so moving to the single cell, then these experiments will become quite expensive. That's why I think at that time, or even now, it's quite relevant to develop methods to guide the experiments. So for example, in the lab, you can only do very limited experiments. Let's say you can try 20 or 50 drugs, depending on the budget that you have. And what you like to do is basically expand this library of drugs by potentially predicting some of them, and then narrowing down the set of hypotheses for the next experiment. This is also very closely aligned with the concept called lab in the loop. And the concept is the experimentalist does an experiment, we'll get some data, we train a model, we predict unseen phenomena in the data. Then based on that, the experimentalists decide what will be the set of next experiments that they would do to potentially improve the accuracy of the model, or to satisfy a certain criteria on the modeling side. And this loop can continue until you achieve your desired result. Hope that made sense. And
it does.
So then what I developed during my PhD was kind of aligned with predictive models to be part of this loop. And today in my lab, we're also doing something similar. Literally extended it to different modalities. So not just predicting RNA or transcriptome, you can also predict, how the morphology of the cells change. And then biology is it's a multi-scale system. So you have the RNA molecules, you have molecular level, you have cells, you have tissues, you have the organs. So the question is whether you can now extend this to higher scales, not just predict what a single cell does, but basically predict how, if you modulate a cell, how the surrounding cell or the surrounding tissue would also be influenced as well, which kind of is a more challenging problem, I would say, because it's inherently a multi-scale setting, and then you have to propagate the prediction. It's kind of like a social network, I would say, right? And let's say you would perturb a node in that social network, and you would like to propagate the effect across the whole network, and then predict it. But this network is a multi-scale network, right? So you have the network of genes within a cell, and then you have the network of cells, and the network of cells and cells, and then it goes beyond this. So since 2017, 18, when I started my PhD, back then when we published our first paper, which was my first PhD paper called SCGen, which stands for Single Cell Generator. Okay. So the paper is from it was published in 2019, right? But then we submitted it maybe a year. It was quite actually a fast process for maybe one of the fastest papers that got accepted in my career, even though the idea was a bit sci-fi back then. I mean, now people are talking about generative models, whatever, predicting this and that. They predict structure of a protein and so on. But back then it was this weird thing, right? So I actually remember for the first time when I presented it in an internal seminar, people are like, this is nonsense. This will never work. And which is, I think also a lesson for me, because sometimes when you do something new, when you do something like that people haven't done it before, and then especially at the intersection of machine learning and biology, where if you really want your tool to be useful for the community, basically you have to be accepted by the biologists that they use these tools. And they have certain standards for what works, what doesn't work, because their mindset is wired in a different way compared to the computer scientists who care about numbers, benchmarks, etc., etc. So these things doesn't necessarily translate to be a meaningful thing for biologists, if you get what I mean. And so the idea was quite simple, the first paper, and the idea was, can we take the gene expression data, we put it into a manifold, low dimensional manifold, and then in that low dimensional manifold, in this case, back then VAEs were hot, right? And then they were a thing, VAEs in GAN era, feels like, ages ago now. So we actually trained a variational autoencoder. We projected the data into a low dimensional space. And if you look at this plot, so the circles are, let's say, unperturbed cells, and the triangles are the perturbed cells. And the perturbation is a general concept here, right? It could be a drug, it could be disease, it could be anything that alter the hemostatic or the normal condition of a cell toward another cell state. And the colors reflect different cell types. And the idea is, let's say if you've seen like the the purple, the red, and also the greenish, bluish cell type, and in two condition, can you actually predict how another cell type, like an unseen cell type, in this case the blue cell type, would respond to this perturbation? And it's just important to know, right, when you apply a perturbation, let's say depending on the perturbation that you do, sometimes they have a global effect. So they basically, perturb same set of genes or similar genes or the same pathway across all the cell types. But they could also have cell type specific effects. So this is the nonlinearity aspect of it. So if you train a linear model, you would be able, or you should be able to predict the global effect. But, generalizing to spatial effects, especially for the cell type specific effects for an unseen cell type, it was something that it was a thing, it was like back then. Even now, I think it's still a challenge. And that's I think what we showed in this paper, that this nonlinearity of the model to learn this complex manifold could help to, generalize better. And there are obviously limitations here. And but feel free to, interrupt, if you have any question or so, we can. This is basically the figure one of this paper. It just reminded me all the days of my PhD. Yeah. And so here, for example, we divided the y-axis in panel F. There are different cell types. The x-axis are different genes, and then we divided them into cell type specific markers. And in this case, the perturbation was a chemical, as a cytokine that perturbed interferon beta. And then we have also cell type specific response as well. And we showed that you can basically generalize for unseen cell types and then predict the cell type specific effects. And this was a proof of concept back then. Let me see if I can find
There's
you.
one thing that's interesting if you look at the figure. So this is one of the very first papers when it comes to single cell generative models, and it's, I think it's one of the foundational papers actually of the domain. So still today, it is Escigen is one of the best models, because I know that because we benchmark we benchmark the models pretty often. Escigen is still one of the best today, and I think it has to do with the simplicity of the model also. The fact that it's a VAE, and then you have linear algebra inside that
So there was like this
latent space.
business. So basically subtracting, and if you remember, Tariq, like back then people were just doing this arithmetics on the faces as well, right? So you take an image of a face with a glass and without a glass, and then we actually took the analogy from that, like, a person with a glass could be on perturbed cell. And then I actually presented this in our internal seminar, and people are like, what is this?
What is this? Cells and glasses.
Because right now people are like, are familiar with AI, right? Everyone is using GPT, ChatGPT. They kind of like, at least it has been printed in them, or like imprinted in them, like AI is a thing. Back then it was not, right? It was just like,
It was, as you said, it was science fiction, and
it was very,
the data set you
science fiction,
yeah. And also the data set you've shown, the Kang data set with interferon, the data set you used is also a very small data set compared to what we have now. So everything was new. And if you look at the figure that you were presenting, you had the benchmarking, and all the models that you benchmark against, if I remember correctly, none of them is a single cell model.
No,
because none existed.
I mean, it was even like, we basically formulated the problem from scratch, right? So we had to come up with like benchmarks ourselves. Like, how would you do this if you didn't have this model? Just like, and my supervisor back then was like, let's do PCA, let's do linear regression, this and that, and see if that makes sense. And then what was quite, like, I think astonishing was, like, we actually showed with the linear models, you can't predict the cell type specific effects. You can actually get away with a global effect, but the other things you can't really predict. And then throughout the paper, we even like went further. I don't know why I did that back then. We went for like cross species prediction, some like really, even still, I would say like sci-fi too many. But that was, I think, one of the reasons. And then we got a bit lucky as well, because we ended up having, I think, reviewers that had an open mindset. And then because most people, if I would go back to 2018, a lot of people will say this is nonsense, right? And so we had I think we got lucky as well that we got reviewers were at least not crazy about
Yeah, and it's true, because if you remember 2018, you probably submitted it around 2018. That was the time where generative models started to become recognized, basically. VAEs and GANs. That was the very early stage of generative models.
And VAEs, but then the applications in biology were quite limited. Now I think it's I think most importantly, it was not about a model because AI is very fast moving, right? But it biology, on the other hand, is super slow, right? So, for example, an AI a year ago is kind of ancient, or maybe 2 years ago is ancient. Whereas in biology, the scale is very different, right? So if you look at 3 years ago or 5 years ago, because it takes time, the wet lab, experiments, data generation, all of these things will take time. And even a paper revision, right? Say if you submit a bio paper to one of these so-called prestigious journals, and a revision round might take a year or two, right? Which is crazy, I mean, probably you and me working on a computational side, we never got it. But there are people who have to go in the lab for 2 years for a revision, and then so
yes. And just to give an idea, in 2018, we were pretty happy. Like when we talk about generative models from 2018, we were super happy to have something that looks like a face that's blurry on the edges,
Yeah.
and you can remove the glasses or change the hair color. And the image were pretty small.
It was blurry. Like you could just see
Yeah.
I don't know, a very shady and then faded picture with some brown colors or whatever. If you change the color to brown, I think.
Yeah, and they were like 64 by 64.
Yeah, that's true. That's true. You could just see an average thing, right? I guess, like, of things. But it's fascinating, right? If you just look back where we were in 2018 and then where we are now, and that again, like contrasted with the biology side of things. And again, like, I think one has to be fair. Like, biology is super complex. It's much more complex than AI, I would say. Yeah, there could be discussion, but like there are like a lot of like uncharted territories in biology because it's so complex, and then there is not much data. You can't really mine the whole internet and train models and so on. It's very complex, and but yeah, I think it's quite fascinating where we are now.
Time also, as you said, it takes, there's time that you cannot compress in biology. So you have to expand your cells. You have, so, I mean, it's just by the nature of things, it takes way more time to validate an experiment, to validate a hypothesis. I mean, you can't really validate an hypothesis. You can only infer. Okay. So, but it takes way more time to do it experimentally in a wet lab setting than to do.
Yeah. And, but after this paper, then I think we, once this was published, then I built upon this and then did a few other iterations with like modeling instead of like doing this linear arithmetics, modeling the whole problem as a distribution matching problem. So basically learning a function that matches, that learns to transfer or transport the control distribution to a perturbed distribution using MMD. And now people like they use optimal transport, a lot of other things to do that. This was 2020-ish, I guess, or 2019, immediately after SCGen. And then after that, I went to Facebook AI, and I think we even went a bit crazier. Like, we basically showed that you can, predict combinatorial predictions, combinatorial genetic and then chemical predictions. That also started like in a wave of papers that you see today, with like, I don't know, Gears, a lot of other papers. And then like now people train foundation models to predict genetic perturbations. For the first time, we actually showed that in that paper that you can, just like predict combination of genetic perturbation, CRISPR perturbations, using a model. And like now there are like a lot of discussions whether these models work, whether you can beat them with a linear model, etc., etc. But I guess, which is not fair, I would say. I can talk about it for some time, but like I think that was just like you could see the evolution of things. And then after that, I moved away a bit from the perturbation modeling. And now I'm very happy. It's a very, very active and dynamic field. A lot of papers are coming out. I think there should be probably more than 50 methods or so. And if you just look at like, I was just looking at the NeurIPS papers that came out today or yesterday for NeurIPS this year. There were like at least 3 papers that they kind of directly address this problem. So the community is very active. There are a lot of like nice work scaling these models and a lot of innovative solutions that people have come up.
Yeah. So are you referring to the Systema paper, Nature Biotech?
You mean for the benchmarking?
Yes, for the benchmarking.
There are like a couple of papers. There's one from folks from Heidelberg, like a benchmark paper that was published in Nature Methods. It's forgot his name. He's at EBI. Emble. There is a paper in Nature Methods. They all like, there are a couple of papers. There's like Perturb Bench, there's Systema, and there are probably more than 10 benchmarking papers. And they all systematically show, I think the conclusion is in some cases, these models can't beat linear models, which I think is unfair because, let's say, and then I can talk about it in detail, but like, imagine that you know a phenomena. For example, you know, if I perturb gene A and B, they would have a synergistic effect. Sorry, they would have an additive effect, right? And I design an additive model that adds A plus B. And that's kind of like the ground truth function, right? And, you know, it's just like, we know this will be additive. So if you design a simple additive model, which basically captures the whole phenomena, you can't beat it at all, right? And so even if you train a neural network, if you're lucky, best case, you converge to a linear additive model, and you would be able to, you know, predict that. And the other challenge is the data is quite messy, I would say, right now. Most of these benchmarks have been done on perturb-seq data sets. So these are like, you know, genetic perturbations, either like, you know, knocking out a gene or activating a gene, like, you know, like CRISPR-A, for example. And so these experiments are inherently biased when folks design them. So for example, if they perturb, let's say, 250 genes, most of these genes were from, you know, selected pathways. And let's say if you perturb a cell with a gene that is part of, like, I don't know, cell cycle pathway, and they selected 5 other genes that they kind of do the same thing. If you learn an average effect for all of them, you would basically get away by predicting an average effect for all 5, because the effects are quite subtle when you do genetic perturbation. And then you kind of like can hack the system and then predict that. But the value is not in predicting those. I would say the value is in the context transfer, and then is where you actually have, you know, like continuous systems. For example, if you have a perturbation that causes, and like before I talk about it, most of those perturbations are actually, they have been applied on immortalized cancer cell lines, right? So those cell lines, they don't differentiate. They're like immortalized, you know, they just perturb them, something happens. There is no differentiation. There is no, you know, there is probably no dynamic or very little dynamic happening there. So for example, if you go back to the 2018 SCGen paper, what we showed that you can take the effect or like uplift the effect from one cell type and then predict it in another cell type, right? So you change the whole context, and that was also in vivo data. So not in vivo, I guess, like it was not immortalized cancer. They were like blood cells, PBMCs, right? And we know PBMCs differentiate. We know they're dynamic, and there is a trajectory. And I think the point is we should make this setting a bit more challenging and a bit more interesting, that these models could actually evolve and then they could shine, because that's where the nonlinearity happens. If you can model the whole system with a linear model, then, you know, so what? Like if you go to the nonlinear system where these perturbations would actually push cells to differentiate differently, and then they will change the trajectory. So there is a continuous effect. There is a very nice paper from Dominic Klein and Fabian Theis called Cell Flow, where they actually shifted a bit from, you know, discrete perturbations and then talk about more continuous perturbations like cytokine, like, like morphogens and organoids. So those are actually quite interesting, right? And I think that would be really hard to, beat those models using a linear model. I mean, there will be always, if you know the underlying function, you can always, come up with an exact model to do that. But I think the value of these models is where you have nonlinearity, where you don't have data, and then on more continuous differentiating system rather than immortalized cell lines. And the fact is, we actually showed this in 2018. So if you go back to the CPA paper, the last figure where we talk about, this paper is kind of like mix of two papers. We had Chem CPA that was published in NERPS 2020. And it's quite funny, actually. This paper was in, we first submitted to Nature Biotechnology back then. And so it was in review maybe for a year and a half. And we even did like a experimental validation, generating a new data set we call like combo cyplex, which is the combinatorial drug perturbation screen that we did. And then we actually showed that you can predict combination of drugs. And then here we showed that this model is, if you look at the differential expression, is it small?
Can you put it in full screen?
Oh, okay. So if you look at here, here we showed that, like, it's, let me see if I can get rid of this. No, I don't know. So here we show that you can actually, on the differentially expressed genes, we can actually perturb, and then, it's actually better than a linear model. And we even showed the cases that this model fails. So for example, for the combinations, that are quite different from what you've seen in the training. Like if you look at panel B, so the greens are the combination that you've seen similar ones in the training. Gray is like, in between, and the orange and red are like, it's getting harder and harder. Mm-hmm. If you look at like, the variance of the predicted effect. So the variance is quite harder than predicting the mean. Yes. Which I think one of the things we should still work on. But even on the differential expressed gene, it suddenly collapsed to 0.6 or so. For some, but I think what is quite fascinating is that here we also showed the concept of the perturbation space. Back then, you can learn representation of the perturbations, and then perturbation with similar functions can be embedded close to each other, which I think could be quite useful for drug discovery, repurposing, and so on. But what I wanted to show is this plot, where we actually showed back in 2018, the linear model are quite competitive compared to the nonlinear model, right? So, and that was because of the data. And I think the whole story was, the funny thing is one of the reviewers actually picked up on this, said like, okay, this is all nonsense. And they kind of like, ignored the whole paper. We showed across like multiple settings, and I think we've seen like, similar plots like this starting 3 years ago in like, 5 or 6 other publications that people are, continuously showing what we showed like, 5 years ago. And, but it's good. I think that the more that we come up with these benchmarks, the more rigorously we benchmark these, because this is something that is very close to the application, for pharma, for drug discovery. The more we explore, the further we try to fail these models, the better it is. And, but I think we're in the right trajectory on the modeling side. The challenge is in the data side. Even with these larger data sets, like there was a data set that came out like with 100 million cells with 300 drugs or so. And again, those are all immortalized cancer cell lines, right?
Yeah.
And I think if you want to get closer, if you really want to convince a biologist, cancer cell lines are, very valuable. Like, people had like Link's data set on bulk harnessing, and has been quite impactful for years in both academia and biotech. But I think we should move on from these static systems toward more like organoid models, differentiating models. And that's where I believe the value of these models will become more evident.
So again, I think we should explain what immortalized cell lines are. Because it's weird because it's basically you take cells from a cancer and then you immortalize them. Basically, you make them immortal. They keep dividing forever, and there you can also synchronize them, so they're all on the same state. But they're also very different from cells that are inside the human body. So they're models. Basically, they're not the real thing.
Yeah, and they're always there's always the fun thing about the it's not the farmer. Like it's like they always like, there is a source for them, and they come always from like a patient 50 years ago. So they took those cells, and then they've been preserving this, and then it's actually a weird concept, no? Like someone who probably died right now, and then their cells are now like it's. But like I guess so, like and that's something that I'm actually actively doing right now in my lab. So pushing these models to more realistic settings, where we're basically generating organoid data, and that's also quite a challenge by itself. Like how to create a mini organ in the lab, and 1,000 things can go wrong. We're lucky to have a lot of like cool collaborators here in Cambridge. They're doing an amazing job. But even like coming up with a system to have a faithful model that you can perturb them is quite challenging. And maybe I can I can just share my screen and show you some of the things that we are doing in the lab.
I think we should also talk a little bit about how the type of data, how it evolved with SCGen. We started with, it was just a single cell RNA-seq. It was also earliest version of single cell RNA-seq. Then we had site-seq, attack-seq. Now we have perturb-seq. So it's I think we should introduce all these different types of data and how it's getting more and more complex, and what we get actually from these different types of measurements.
So there are like a few major players in the single cell domain on the data generation and technology. I think one of them is like 10x Genomics. So 10x Genomics, most of I think commercialized. They kind of had a monopoly for a few years, but now they're like Parse Bio. There are like other companies that they're basically also pioneering global single cell kits. So it's getting cheaper and cheaper. So you can generate 100 million cells with a price that was unbelievable in 5 years ago. So you could have not probably done these experiments back then, but the technology has evolved. It has gotten cheaper, and then sometimes also the quality has improved as well. And so people are quite, I think, coming from 2018, there were these well-based technologies, and then later, right now we have Flex. They're like a cheaper version of the 10x that you can run on archival samples, and where we have a lot of data in archival samples, cancer data. A lot of patient data is actually archived using. Basically, they store them in a way that you can preserve them. If I want to simplify it, and there are different ways you can freeze the samples, or you can coat them with a specific chemical and structure, and then they can be preserved. So anyway, there are technologies right now that you can do it way cheaper, and then you can multiplex them as well, meaning that back then, if you would have done 10 experiments with 10 different perturbations, oh look, that's funny, with 10 different perturbations, you had to run them separately. That's why it was quite expensive. Right now you can just pool them together in one experiment, and then at the end you can demultiplex them, meaning that you can just tease out or discriminate a cell based on whether the cell was perturbed by drug A or drug B and so on, while at the beginning you just pulled everything together and then extracted the RNA information. And I think that the other big change that has happened since then is the rise and also the maturation of the spatial technologies. So back then, I think the first single cell experiment, if I'm not mistaken, was done in 2009 here in UK. I think in LMB, which is in Cambridge, and I think they profiled, if I'm not mistaken, or maybe first or whatever, second or so, that they profiled, like, I think they captured 9 cells, and then they later expanded across, like, they kind of scaled it. And then now we have data sets with 100 or 100 million cells, or more than that. But what we what we were not able to do back then was to retrieve not just the RNA, but where that cell was in the tissue, right? So we basically, the spatial information was lost, which is crucial, right? Because cells, they don't operate in isolation. They talk to each other. They localize depending on where they are. And in a recent paper that we had, it's a generative model called MinFlow. We actually show that you can decompose, so that the cell state or the RNA or the gene expression in a very simplistic manner. Let's say we can divide it into 2 vectors, a vector that is intrinsically driven by the cell, or proportion of the genes that, irrespective of where the cell was in the tissue, you would have had these level of expression. Again, these are all our assumptions, right? In reality, they could be very different. And then you also have a component of the gene expression that is induced by the environment or the microenvironment. That's what we call it. And so if you'd be able to tease out these 2 different effects, then for example, the difference between a T cell that has infiltrated the tumor versus a T cell that is just sitting there in a standby mode, right? Because they get activated and they change. And so I think now we have ability to extract that information in a scalable manner. And there are different technologies. So for example, you can have a slice of the tissue, you can image them, and then based on the segmentation that you get, you can where each cell was in that 2D grid. And there are also other technologies, sequence-based technologies, that you try to kind of attach an identifier to every cell and every location, and based on where they were, you had unique identifiers of different X and Y's in the tissue, and then you can assign them to where they were in the tissue. Irrespective of that, we have these technologies right now, which I think would be, maybe in a year or 2, I think we will get there. No one will do single cell RNA-seq anymore. Everyone will do spatial, because it's just that is the exact biology that you have to see, right? And that give you much more information, and we didn't have that information back then. So then it will become quite interesting now. Then can you perturb not just a single cell, but perturb the microenvironment, and then predict the effect on every individual cell in that microenvironment? So for example, what we had in this paper that we put out, MinFlow, is we kind of simulated different spatial perturbation. Let's say you have a microenvironment, or microenvironment is a fancy term for the community of cells, right? Some cells that are sitting together. We basically questioned whether if we can actually remove certain cell types from the cocktail and then predict how other cells will be influenced if this cell type was not there. Right?
Yeah, because cells talk to each other.
Exactly. This is kind of depletion of the cells, and then see the effect on others, right? And this phenomenon happens quite a lot, many drugs do that, right? They inhibit certain cell-cell communication, or they kind of promote certain cell-cell communication, or they deplete cells or whatever, right? I think there are different ways. And so the other thing that you can test with these type of models is if you add a new cell type to the cocktail, right? And in that paper, we showed we try to mimic a cell therapy for atopic dermatitis. So there's eczema, and even now in 2025, we don't have a treatment for eczema, right? And then one of the potential treatments is T cell therapy, right? But to really test that, it's really challenging. You can't do that, and you need to have very sophisticated organoid models that you co-culture them with T cells, and you might see something, or you can't really do that in human as well. I mean, FDA right now, I think they lifted the animal experiments right now. So it's just getting easier for folks to, further use these models. But I think this kind of also shows the value of these models. You can simulate phenomena, or like you can assess or evaluate hypotheses that would have taken ages to assess. And because
and when you say models, it's AI models.
Exactly. I think this is, for me, this is what drives me right now, and then what we're actively doing in my lab. And I have to say, like, if you compare it to what I was doing 5 years ago, it's much slower, because I mean, I'm just going back to what I told you, like at the beginning of our conversation, of like, the biology versus the machine learning. So there is a spectrum. If you want to, at least in academia, publish papers that are more like methody, and then, I think almost everyone beats other models in their papers, right? So that's kind of like a joke right now. Everyone publishes methods and they outperform every other method. So it has become kind of like, and it sometimes become like meaningless, right? Okay, so obviously you outperform other models. But what we're doing right now is, like, I think I myself, also the direction of the research, is getting closer and closer to the lab to see if we can really, simulate a known biological system, predict unknown phenomena, and then try to really replicate it in the lab. And that is much slower compared to the type of papers that I was writing in my PhD, right? Which are like more methody. And the further that you go toward this, like, in this spectrum from the method to methods really, discovering something, or like, doing something non-trivial, it gets slower and slower. But I think the long-term impact would be much more down the line for drug discovery. If you just be able to simulate those spatial perturbations, there are a lot of implications for, cancer, for, cell therapy, for treatments. You can simulate many of those.
Environment. So you were about to show slides about organoids.
Oh, about organoids.
Yeah, like
I mean, they were not. Say it again. Sorry.
It's super interesting, but I know you have meetings afterwards, so I have to give you enough time basically to show like the most interesting stuff.
I think I can quickly show you something that, like, this is still like work in progress, but so hopefully we will have a preprint out. But I mean, right now we're talking about, like, we're like, it's 5th of November. So the PhD student promised to share this, to basically put it out before the end of the year. So let's see. Usually never happens, but.
I also have a deal like that with students.
Yeah. So, but I'll show you, just an example of what we're doing right now. So the idea is, do you see my screen?
Yes.
Okay. So the idea is that can we actually come up with models that take a cell from a source condition toward a target condition, and then predict the effect, right? This is very similar to what folks have been doing, what people have been doing on natural language, right? So the backbone of many models that we have are, like, encoder decoder models that you feed in a sentence in English, and then you have a sentence in German, and then the generative model spits out the translation there, right? And the idea is whether we can do a similar thing here with cells. Can we put in a source cell type, and then predict how this cell will change in the target cell type? And once you learn this transition, then what you can do is, let me show you this. What you can do is basically perturbing the system. So let's say if you have an initial cell state, you can, overexpress IL-7, and then predict how the cell will change, but not just predict the end state, but also predict what happens in between as well. And so these are like, this is one of the examples that we've done in this paper. Let me see if I can show it to you here. And one of the challenges that we have in the lab. This is in collaboration with a colleague here at in Cambridge, Ms. Hanifa, and she's a dermatologist. And again, like just this is work in progress. A lot of things might change. But the idea is that it's quite challenging to actually make artificial skin in the lab. And the challenge comes from like, you know, there are like different aspects to it. But one of the challenges is when you try to take cells from a patient or from a sample from a human skin and try to, you know, differentiate that, or you start from stem cells and you try to differentiate that to become like a skin organoid, where you have hair follicles, where you have like, I don't know, like a proper skin with hairs with different layers and so on. One challenge is they won't necessarily mature. So and it's just like a very complex process. So if you start from like, you know, stem cells and then you differentiate them, the current protocol to get to a workable skin organoid is a 19-week process. So that's why, you know, it's slow. Mm-hmm. You have to wait for 19 weeks to see a result, whether what you cultured in a lab to make an artificial skin is a legit thing or not, right? And this is also a dynamic process, right? So you start from day zero, cells change, a lot of new cell types will arise. And so a lot of things will happen during this 19 weeks process. But in the end, in most cases, and this you can generalize this to other organoids as well. If you know, I know, culture brain organoids, or gut organoids, kidney organoids, et cetera, they're not perfect, right? And so they don't look like a mature human skin. So that's the first challenge, how to get them closer and closer to the human skin, adult human skin, or even like, I think at this point in week 19, it's not even adult. It's like, it's very close to the fetal skin, to the skin of a newborn, for example, right? And even it's not like that as well, right? It's not like a human skin. So it's somewhere in between. It's like a hybrid, right? So the first challenge is how to mature them. The second challenge is there are a lot of side effects. So there are some cells or, you know, structures that they should have not been there. And they're like, you know, for example, there are cartilage cells. There are like, I don't know, just to make it simple, there are like cells that you don't want to be there, but they're there. Mm-hmm. And so the question is, how can we optimize this protocol? How can we better generate more realistic skin organoids or artificial skin in the lab? What we did here was we trained this model that I just like highlighted and showed to you on this data. So we basically learned just for the context, we pre-trained this model on like 120 million cells across human development. So it has seen some other phenomena in development, but what happens is this model received the cells at like, let's say week 1. It has to predict what will happen to them at week 4, 7, 12, whatever, 19, and so on, right? And so when I say predict, given a cell from week 1, you have to predict how the cell or gene expression would change in week 4. And then you do it simultaneously across all of these time points, and that's how the model actually learns it. Once we've done that, what we did was we basically went back to the week 1, and then we did an in silico genome-wide perturbation screen, meaning that we perturbed every single gene in week 1 in all cells, and then we simulated if these genes would have been perturbed, how would they change the differentiation of the cell up to week 19. And what you see on the right side is what we call a perturbation map or a perturbation landscape, which is the average effect of these perturbations across all of these 19 weeks. So if 2 dots are embedded close to each other, it means that these 2 perturbations predicted by this model had this similar effect. So this is kind of like pseudo-bulking the synthetically generated single cells upon the perturbation. And then we clustered this. So when you see these colors, we organize them, or we group them into different functional groups. For example, when you see cell cycle, these are the genes that modulated the cell cycle throughout the organoid development after the perturbation of these genes. So what we did next was, okay, what we want to do is we want to pick some of these genes that actually mature cells, right? That would just push these organoid cells to become closer to the fetal skin that we have from the fetal samples that were published previously, right? Because those are actually, could be ground truth for us. So what we did was we defined set of markers that you see on the dot plot on the right side. So these are markers that are markers for maturation of mesenchymal cells, like fibroblasts and so on. And these are quite important cell types in skin. So what we did, we wanted to see each of those points that, sorry, the dog has gone crazy. She's using me now. And each of those points, if any of those perturbations can modulate those genes that we are interested in. So if you in silico perturb that gene and then predicted the outcome, is the outcome aligned with the modulation of those 5 genes? If those 5 genes have been modulated or upregulated. And that's how we scored those 5 or whatever, 6 different clusters on the U map that you see on the left side, and then rank which set of genes can potentially modulate these gene program to push cells toward maturation. And on the heat map, you see that there is a cluster called Wnt regulators, and you can see the gene names on the top on the UMAP as well. So these set of genes are more likely, at least these are all predictions, right? They're more likely to modulate this gene program, thus they might further mature skin cells. So what we did, what you see on the box plot here, we narrowed down the hypothesis into one of those genes that were prioritized by the model. This gene is called GSK3B, and this is part of the Wnt pathway. And Wnt pathway is generally used a lot in in cell growth and iPSC and organ modeling, and in some cases it can promote growth, but it's a very general pathway. And what we found out, this gene is somewhere in that pathway. I'll just make it very abstract here. And that can there's upstream of the Wnt, and that can modulate the Wnt signaling, right? So what you see on the right side on the box plot is prediction of the method upon the GSK3B, i.e. the Wnt activation at day 29, day 50, and day 131. So when you see day 21, the previous day here was day 6. So what we did, we actually perturbed the cells in silico in day 6 and then predicted what will happen to them in day 29, 50, and 131. So first, the expression of the genes will go down, and then upon the day 50 and day 131, they'll increase, at least in silico. So what we did then, we went back to the lab and we tried to simulate this. So we took the skin organoid, we actually found the small molecule that target the GSK3B gene, and then treated the organoid with this, and what we saw was quite astonishing. So basically, in the validation, we saw that the pattern that it first goes down and then it will increase. And this is still work in progress. We have other hypotheses, but this can show you, how using AI you can facilitate experiment design, right? Because to be honest, doing this, if you talk to the biologists, it's a very brute force thing. There is someone who's a specialist in skin organoid, and they really know what they're doing, and what they can say is let's go try this gene. And if you imagine how unlikely it is, if you select a gene, among, tens of thousands of other genes, and the chance that it work is, it's small, right? And then here, literally in 2 weeks on the computational side, we perturbed everything that we could. We could have even done the combination as well, but the challenge was the validating the combination is quite challenging as well, right? So you have to, the guide design and how you do them. So usually when you perturb genes, the ideal case to validate them for us in the lab is if we can find a small molecule, instead of really going to do the perturb-seq, we can find a small molecule that targets this gene, because then treating the cells with the small molecule is way easier compared to perturbing the gene. And I think that's why I'm quite hopeful. And then, and there are ups and downs in our research that it took us a long time to get there, to have that thing working. And I think there is a lot of potential, right? So imagine if you do these things on, instead of maturing the organoids, do we know how to, steer organoid toward a disease-like state, and then do a drug screening to see if we can reverse the skin disease toward the healthy. We're not still there. So, for example, we're actively working on it in my lab in collaboration with our partner to, prioritize candidates that if you treat the organoid with them, it basically pushes cells toward the disease cell state rather than. But it's just step by step. Yes. First, you have to mature them, and then.
I think this is the new what we were saying before, SC was science fiction back then. So this is the equivalent now.
Yeah.
Now
I'm a bit afraid to submit these papers, because I think they would be able to have resistance. And then we have a very similar paper for the Spatial now in review in Nature, and they usually have a biologist to review it as well, and they kind of freak out, right? So I think our only chance to really, convince them is to do more and more of these experimental validations. And most people, they actually pick it up, there is a value in this, because otherwise it would have taken ages to do these experiments or come up with a hypothesis like this. So the value is not replacing the experiment. The value is to guide you to do more efficient experiments and then facilitate the discovery.
Yes, I agree. I think AI got somewhat of a bad name because of the PR of AI. There's always this idea that AI is going to replace people, but this is not about replacing anybody. This is about speeding up the hypothesis generation. Yeah, which is basically one of the big bottlenecks.
Well, Tariq, postdocs, or even all the seminars, because there is a big angst between in the bio community that what will happen, right? And I would say, at least this is my opinion, it could be wrong, or it could turn out to be wrong, but I think AI is let's say, I don't know, people had 30 years ago, how computers changed how we analyze the data, how Excel, for example, changed compared to drawing things on a piece of paper and calculating all of those things. This will be very similar to a calculator or whatever. This will become a tool as well. It all depends how you want to use it. And it will just the more proficient or the more efficiently you use these tools, I think your research will also become more efficient as well. So every biologist, I think in a few years, they have to know how to use the systems. Otherwise, I think that the way of doing science will also change drastically. Again, these are all predictions. I could be wrong. In my opinion, they will drastically change in a few years. The way that we design experiments, the way that we do experiments, the iteration between the computer and the lab, and so on. And I think we have to be prepared for that. We have to educate people for this, and also kind of remove this angst that this is not going to replace the experimentalist. This is just another tool that you have to work with.
Yeah. I think it's the best tool for the experimentalist. I think it's as an experimentalist, you don't want to go and spend 15 years doing all the possible experiments. So you want to focus on the most likely experiments that will lead what you want actually. So and that's the
And I
whole
must
value
say,
of
Tariq, like
these
we
models.
might not be there yet, right? At least in our domain, we're not there yet, right? We don't have perfect
No.
models, or like there won't be perfect models, but right now I think this is still work in progress, right? And then a lot of talented people in the community are working on this. So we are pushing toward that direction, but it's just a matter of time,
Yes.
right? Whether
I just don't want to scare the biologists too much, but you're right. I mean, what we do, which is AI applied to fundamental biology, is very slow compared to for example, happened imaging and video or NLP. We're very, very far from being able from a ChatGPT level, basically, in biology. Yeah.
I think that is also something that we have to be very careful about, because the way I think it's just like, I think we're all culprits in this, that like the way we advertise these papers on social media and on Twitter, X or whatever, right? It's people come up with these, I would call like a nonsense term, GPT for biology or whatever. Like, I think this will actually be harmful for the community, because if you hype it and then people try it and then see this is not, this is not living up to the hype that the authors or us, like I've done it myself as well, like we're all culprits. We have to show reality, show these things work, so we're in a certain direction, and we also show the failure modes as well. I think the challenge is, in the end, in academia, the currency is publication, right? People are promoted based on their publications. They get like, leadership positions, et cetera, based on the publications. And then, so many people are afraid to show failures in the publication because the reviewers' reaction could be quite negative, right? If you show, which I personally, when I review papers, I actually ask the authors to show the failure modes. And I find it quite valuable, because that would be the thing that the users should know, right? They should know this tool is usable in this, it won't work in these scenarios. Rather than like, I think like, it's not a negative thing for the method, because the method, we know every method has limitations, right? And then we have to transparently show this, but the community also has to be receptive. Both authors, reviewers, grant writer, reviewers, and so on, they have to be receptive to this, like, this is not going to do magic. And it's not going to solve everything, right? It does something right, and then it does 1,000 other things wrong. But that's the nature of science, right? We have to go step by step. And I guess for the AI, at least from 2020, like, I mean, right now, at this stage that we are, we have to be very careful in terms of wording, how you communicate this. Because if you just overhype things, if there are other words that they actually do it, then people will not believe it, because there are like 10 other papers that they claimed it. And it might be the cycle of science. I don't know, but this is just...
it's the cycle of AI.
Yeah.
But I think the issue is that we went from it doesn't work, it will never work, to it's biologically solved. So by biology solved, and that's I think it's and the problem is, it's not that people won't believe it. I think the problem is that people will believe it now, and then they test the models, and then it's not up to the models are not up to the par yet. And then you lose confidence, and
at least that's what we're seeing in our community, Tariq, right? So if you look at the number of, like, you can divide the papers that are coming out in a single cell community to data papers, or like the discovery papers, or whatever you want to call them, and the method papers. And if you go back to if you look at I don't know, I haven't looked at it in detail, but just go back and look at all the papers that were published in 2025. How many of them have used foundation models? I think there might be less than like your fingers, right? And they're still using SCBI, SCRHs, all of these things, because they work.
SCGen.
Like all of, exactly, SCGen and so on. They're like ancient papers, because none of these things are systematically tested. they're not working in many cases because the speed and the race, and again, this comes from the AI kind of mindset as well. Like, people will just like, back to back publishing papers, improving stuff. But I think the reflection that we're seeing on the usage and, where actually is anyone actually using a foundation model to build a single cell atlas?
I mean, people are trying to,
but it's,
but
so I think these are the questions, like, when we, I guess all of us who are working in this domain to, again, make it usable, documentation, and I think we're getting there. They were like, the codes and everything are getting much better. It's just the robustness and the usability and whether they actually apply it in the real case scenarios and real examples. Yes. And I think we should be more,
Yeah, and also think there's the data is fundamentally different. When we talk about biology, it's not like NLP, it's not like images. You can just copy paste the model and then hope it will work on biology. Like one example that's I mean, we can take protein structure because proteins are, I mean, it's one of the big things, one of the big wins, like in biology, the recent ones. If you take, for example, if you say a protein is a book, and you apply it like naively, you apply the same models that you would apply on an LP, you apply it to a protein, you rarely have a situation where in a book you can change one letter or one word and it becomes a completely new book. You have that with protein structure. And when it comes to single cell, we don't even know. Because we're just getting the data. It's like with all these new sequencing methods, with like we went from in span of like a few years, biologically, it's like 15 years, we went from bulk sequencing, which is basically getting an average of all the cells, like all the players, to single cell sequencing, which is like sequencing the players, which is now spatial single cell sequencing, which is sequencing the players and knowing about the field. And before that, we didn't know. So we don't even know what's like it's a surprise thing, basically. It's like we getting the data as we speak, like so we don't even know what makes it hard. We have to figure that out.
So but I'm quite hopeful. I think like in the next few years there will be a lot of like cool things that AI will do for science.
Me too. I'm super hopeful about that. But just an explanation of why we're not at, for example, a Midjourney level in imaging or a ChatGPT level in an LP, because we are discovering the biology as we speak, and we are discovering what's important as we speak.
Yeah, and then careful validation, and then like I think one thing that also has changed the way that I also do research is, because our institute, I'm one of the few people who do AI, does AI here at Sanger, right? And you're so close to people, who are doing like, proper biological discovery. And then you can also get a sense, what is relevant for them, what do they care about, and a lot of I think cool ideas, hypotheses, and inductive biases that you can put into the architecture design for AI models, it can directly come from them. So I think for us, sitting down with them, talking to them, and like, for example, the moment that I came to I started my job here at Sanger, right? I was doing a spatial method, like it's called Niche Compass, was published in Nature Genetics a while ago. And so the idea was to identify tissue microenvironment, tissue niches, and characterize them. And later, right now we're scaling those methods to train larger methods. And then a lot of people are doing this on the mouse data, mouse brain. Well, it's not that like mouse biology is not important. It's like the main tool that we're using right now. But I think the shift that I think from one of my colleagues is let's just do it on the human, and that mouse brain is a very structured setting, and even brain itself is it's very layered and then spatially well organized. So it's very easy, to not on the function, but rather on the spatially map it. and now we've moved to the human data. And if I did not have exposure to these colleagues, I would have done a very different thing. So I think for us who develop this type of tools, being in a direct contact and day to day, and many people might not have it, they might not be, in a certain environment that I am, but you can still reach out to people and then talk about or ask their opinions, whether what we're doing is relevant if we care about it. Because again, you can publish things. That's not a question, but whether you publish something or you just, design a tool that is useful, that has a very different, I think, process compared to just a paper. And, which I think, it's also a longer term process. It's painful sometimes, you hear stuff like, this is irrelevant. I don't care about this. What are you doing? It's like, but I think we have to hear those things also, and yes. My other experience is they also have a different mindset. And so there are some positive things and there are some things that again, I like the computer science mindset, to be fair, is that biology is also sometimes very hypothesis driven. And the fact that we make it hypothesis driven on the AI side will not work, because in AI, the more general you learn things, it would be more useful downstream. Obviously, specialized model will work, but I think if you learn a general concept of the thing, it would be much more useful. So I think for us working in this direction, we have to take the validation, the hypothesis generation, and a lot of cool things that we can bring in from the AI side, and then combine this mindset. And one of the challenges that I also have is if you want to do these projects, the people who are in these projects, for example, I have MD PhDs, I have wet lab biologists in the team, biologists, computer scientists. At the beginning, it's a mess. They can't talk to each other. They're like, what the heck this guy is talking about, or this person is talking about, right? And so just finding a common plane for them, and then having this dialogue that they talk to each other and they understand, okay, I can't do this project just with my own expertise, and I need this other person, and try to synergize it. That's a bit more on the management level and also project management, but that's also quite a big challenge compared to, just 3 computer scientists running things on GPUs, trying to optimize it and so on. This is a very different type of project. And I think which is a challenge by itself for us to learn how to work within a multidisciplinary team.
I think but as you said, I think it's mandatory. If you want to have if you want to have impactful work, you have to talk to biologists. You have to learn about the biology. You have to learn about the lab setting. All that is super, super important.
You've been in Chloe's lab, right? I think you also experienced that compared to, how they think, how let's say folks in Montreal would have taught about the same thing. It's a very different thing, right? I think.
Yes. So so my story is a little bit different because my whole PhD was in an immunology lab.
Oh, okay,
so it's I dived straight in. I was surrounded by biologists.
It's very different because I did my PhD in a very it's a dry lab, right?
Mm-hmm. Yeah, it was a wet lab. I had a co-supervisor, bioinformatician, but I was sitting physically with biologists.
Wait, that's amazing.
It was that's how I learned all my biology, and that was my big culture shock was actually the PhD, and so because it's computer scientists and biologists think very differently, and they see problems differently, they approach problems differently, and this is something that we don't realize, but it's when you get to the after the PhD level, there has been the fork is basically high school. So you haven't met a biologist since high school, basically. So you went to 2 different, and getting back together at that level is pretty challenging because you had very different life stories, very different ways of thinking have been ingrained in you.
Yeah, and that plus I think it's also the scales again, like maybe like me moving from the PhD to the PI. It's the expectations that people also have about these projects are quite different, because again, like CS people are quite used to these like short-term projects. Like you basically have these like few conferences spread across the year, like every 4 months or so. Whereas here, like, a proper computational biology like method paper will probably take 2 years. And so it's like a delayed award, right? And then it's like people have to, and there are like a lot of ups and downs,
Yeah.
It's very different.
That's a challenge.
It's a very big challenge. No, I mean, like for people who are used to publish quite frequently and they come here and are like, oh no, it's like the first 6 months.
Take it slow.
Yeah.
That's true. You have to convince students that they have to take it slower.
And take it slower.
Rebuilding this,
And this also requires certain characters, right? So certain characters that are used to these long-term projects, and they can manage day-to-day activities, and I think things can be much more efficient. But like right now, this is the case because it takes time, right? But that's also one of the challenges running a lab like this.
I mean, one of the main challenges.
Definitely.
But and you touched upon like a very, very interesting point. I mean, very true is the way we approach the problems is basically the opposite of the standard way of approaching it in biology, because biology is hypothesis driven. So basically you have you're targeting one thing. But in our case, we can't do that with AI. We never work with AI. You have to take the broader data set that you can get, integrate that, and then derive your hypothesis from there. So it's a completely different thinking process.
I agree. I think this is, there are different ways to kind of like probably, analysts. Right now, we also decided that we also publish in conference papers. Like, we just put some of the methods in ML conferences. So for people to have also a bit of like reward for what you're doing, and then later, convert it to a journal version of it. Because we have to innovate on both sides, right? It's not just the method has to be novel. The biology that we discover, if you really want to, claim that you found something, it has to be novel as well. So it's even more challenging compared to just the data paper as well, right? So because they have the data, but even if you find a new biology, I myself would like to publish method data papers, not just data papers, right? I mean, I might publish data papers, but then I would still like to, that's what I'm doing. That's what kind of motivates me. So we have to be novel on the other side as well. So how you manage these projects to be, not scooped while still keeping the novelty is another thing. But I guess like in most of these papers, I think down the line, the depth and the breadth of these papers will be very different from like, because for our domain, right? It's like Tariq, you just open bioarchive and then you'll see, oh, there is a paper that did exactly what you. So I think we have to move beyond that and just go a bit deeper into.
Yeah, I think so. And managing reviewers is also challenging.
It is.
I don't know how you do it with machine learning and AI conferences. I usually, when I send papers to these conferences, like I think the last one with Generative, it's usually like very dry papers. It's mathematics, basically it's math.
That's
it's
what we
like
also
very
do.
dry.
We put like one or two probably like very shallow analysis that to show like, this might do something. But if you go beyond that, the reviewers won't get it. And then like with the way that people review these things also, they just put it into ChatGPT, and then, it's GPT writing papers, GPT reviewing papers, and so on. So I think for us, it's like we strategize we just put like very shallow use cases that clearly highlights the impact or potential of it. And if we go deeper than that, that's not relevant.
It becomes counterproductive.
Exactly.
Same thing. You also have like, on the other side too, it's easier at least in my opinion, it's easier to get it right with journals, because there has been a history of papers that look like that, but still it can be challenging
It is challenging, right? Because like usually these
reviewers,
journals, they also have like, bio gatekeepers, and these people like you have to write it in a very general way that they also understand it. And most of the time, because of their background, they don't necessarily care about the impact of the method and what it could, what it can do. They just like merely focus on the discovery side, because some of these things, right? Like when AlphaFold came out, right, back then a few years ago, well, like there were predictions, right? But then like the discovery aspect of it, was not yet there, right? And then people have built upon that. Now the design it has become like a normal thing in the protein world, right? So they design stuff and evaluate it and so on. I think for us, again, we have to review this paper with an open mind as well, not just looking the exact thing, but also the impact as well. Yeah. Cool.
That's true.
Great.
Thanks a lot.
Thanks a lot, Tariq, for having me. I think it was good to catch up.
It was really fun. Yeah.
We should next time you're here, just let me know, and then I think I should come, and then
sure.
The Morocco is my
come first. It's no, as we're recording, it's November, so you should you should come here. Yeah.
What is it temperature?
And I can come in summer. Oh, it's great. It's the best time to be in Marrakesh. It's maybe 30 max, 25, 28. It's very dry. It's amazing. Yeah. That's why I say you should come now. I come later. I'll go to the UK during the summer.
Yeah. I think so UK. I think summer is like June, July is the best.
Yeah,
Great.
Definitely.
Thank you so much, Tariq, for having me.
Thank you, Mo. I think we should do it again at a certain time. Yeah, me too. Super interesting discussion. Always very happy to talk about single cell.
Yeah. It's fun.
Yeah.
Great. Thank you so much.
It's fun. And lab in the loop.
Yes, lab in the loop. Yes.
Yeah.
Thank you so much.
Thank you, Mo.
All right.
See you. Bye-bye.